Welcome to Biology at Priyajit, my dear students. Today we explore the cytoskeleton, the cell's internal framework that gives shape, strength and movement to life. Much like the steel structure of a building, the cytoskeleton supports the cell, organizes its contents and enables transport and motion. The cytoskeleton made up of microtubules, microfilaments and intermediate filaments, and they are powered by motor proteins. This dynamic network or structure plays a key role in cell division, intracellular transport and cellular movements. Let's break it down simply and understand how this microscopic framework keeps the cell alive and active. Introduction. Just as the skeleton supports the body of vertebrates, eukaryotic cells possesses an internal skeletal framework called the cytoskeleton. The cytoskeleton is a dynamic filamentous network of proteins present in the cytoplasm. Basically, it provides structural support, cell shape, cell organization, intracellular transport, cell motility, cell division, etc. So the cytoskeletal filaments are protein polymers held together by weak non-covalent bonds, allowing rapid assembly and disassembly. So this is the image showing the cytoskeleton of a cell. These are the microtubules, green colored structures, there are microtubules. The blue colored structures are the microfilaments, and the red colored structure, maroon colored structures are the intermediate filament within a cell. So how do we study the cytoskeleton? There are some advanced techniques that are used. Number one is live cell fluorescence imaging. Second one is in vitro and in vivo single molecule assay. Third one is the FRAP technique, fluorescence recovery after photobleaching, which is used to study filament dynamics. Components of the cytoskeleton. The cytoskeleton is basically composed of three major types of protein filaments. Number one is microtubules. They are long, hollow, unbranched cylindrical tubes. They are composed of tubulin protein. So they are the thickest cytoskeletal element. Now microfilament. Microfilament are thin, solid filaments made up of actin. They are often arranged in branching networks. Intermediate filaments, they are strong rope-like fibers. They are made up of various types of proteins. They provide mechanical strength to the cell. So what are the functions of the cytoskeleton? First one is the structural support. It acts as a dynamic scaffold, so maintains cell shape and resist mechanical deformation. Organelle position. Cytoskeleton helps organize organelles inside the cytoplasm in a proper location. It is very important in polarized epithelial cells. So organelles are arranged from apical to basal ends. Intracellular transport. Cytoskeletal filament act as tracks for movement, such as examples include transport of mRNA, vesicular transport from endoplasmic reticulum to Golgi body, neurotransmitter transport in neurons, and microtubules also serve as tracks for peroxisome movement. Cell motility, they enables movement of whole cells and cellular components. Examples: single dead organisms, the crawling, cilia and flagella; multicellular organisms such as sperms, WBC, fibroblasts, growing axons, etc. Cell division, cytoskeleton also forms the mitotic spindle. They are essential for chromosome separation and cytokinesis. So they functions during mitosis and meiosis. This is the cytoskeleton. Three types of cytoskeleton are there: microtubule, microfilament and intermediate filament. So now we go for the microtubules. We should know the basic structure of microtubules. Microtubules general features include it's a hollow, rigid cylindrical structure present in almost all eukaryotic cells. They are essential components of mitotic spindle, cilia and flagella. Structure of microtubule. Microtubule diameter is approximately 25 nanometer and wall thickness is roughly 4 nanometer. Wall composed of 13 protofilaments. So each protofilament consists of alpha tubulin and beta tubulin. These are formed by tubulin heterodimer. This is a spontaneous binding of the two tubulin. Polarity of microtubules. Microtubule polarity. Tubulin dimers are asymmetric, giving polarity. There are two ends. One is plus end and another is minus end. Plus end is the growing end, and minus end grows slower. Their polarity is very much crucial for directional growth and motor protein movement. Microtubule organizing center. Microtubules assemble rapidly at specific sites called MTOCs. Most well-known MTOC is the centrosome. Steps of assembly: nucleation, it occurs slowly, and elongation, it occurs firstly. So all MTOCs contain gamma tubulin. It initiates microtubule formation. Microtubule associated protein or MAP. They are accessory proteins bound to microtubules. They functions, they stabilize microtubules, they promote assembly and maintain parallel alignment. So MAP activity is regulated by phosphorylation. This is the microtubule associated protein, and they regulate their activity. Tau protein and disease. Tau is a map found in neurons. The excessive phosphorylation of tau protein causes neurofibrillary tangles. This is the tau protein which is associated with the microtubules. Microtubule is also associated with Alzheimer's disease, FTDP 17 dementia, etc. A FTDP 17 dementia is a frontotemporal dementia linked with Parkinson disease, and which is found in the chromosome number 17. The gene is located in the chromosome number 17. It is a neurodegenerative disorder, dementia disorder. So tau gene mutations confirm its role in neurodegeneration. Acha. Now we should know the motor proteins on microtubules. Motor proteins, what they do, they convert ATP energy into mechanical work. So two major types of motor proteins are kinesin and dyneins. Now go for the kinesin. They're discovered in 1985 by Ronald Weil. They present in all eukaryotic cells. This is the structure of the kinesin molecule. It belongs to the kinesin like protein family or KLP family. Their structure, it includes tetramer. There are two heavy chain and two light chain. This is the two heavy chain and these two are light chains. The regions, two motor heads are there. There are these two heads which acts as ATPase. This is the neck region, and this is a stalk and cargo binding tail. This is the cargo binding tail. So function, it moves towards the placent and known as placent directed motor protein. It speeds approximately 1 micrometer per second. It moves in discrete steps at low ATP levels. Dynein protein, it was discovered in 1963 in cilia and flagella. The cytoplasmic dynein discovered later. It is present throughout the animal kingdom. Its structure is a very large protein, approximately 1.5 million Dalton. The two heavy chains plus intermediate and light chains are there. They include the structure. So the large globular heads act as force generators. Function, it moves chromosomes and positions spindle during mitosis. So minus indirected motor, they position centrosome and Golgi component. They transport vesicles and organelles. This is the dynein protein. There is a cargo interacting domain, and there is a domain that interacts with the microtubule. This is the stalk region, this is the motor domain, and this is the head. So this is the interaction between the microtubule and dynein and kinesin protein. Intermediate filament. General features include their diameter is about 10 to 12 nanometer. They are found in animal cells. They are strong, flexible, rope-like fibers. Their characteristics include they provide mechanical strength. They are abundant in neurons, muscle cells, epithelial cells, and they are chemically heterogeneous. So approximately nearly 70 genes in humans are responsible for intermediate filament formation. Structure of intermediate filaments. Their subunits contain a central alpha helical rod domain. They are linked to other cytoskeletal elements by plectrin protein. Plectrin protein also connects IFs, intermediate filaments, with microtubule, and intermediate filaments with microfilaments. This is the structure and assembly of the intermediate filament. This is the image showing the intermediate filament structure and assembly. Microfilaments. This is the image showing the assembly of microfilaments. The diameter is about approximately 8 nanometer. It is made up of actin protein. The actin monomers polymerize in presence of ATP. Properties: two standard helical structures are there which are called as F-actin. So the filaments are polar. When ATP is converted into ADP after incorporation, then majority of filament contains ADP actin. motor proteins which are associated with microfilaments called myosins. So general features include they are first discovered in skeletal muscles that are present in animals, plants, protists and non-muscle cells. Their structure include a head domain which binds actin and ATP. They have tail domain which is highly variable. It contains light chains. Types of myosin: there are conventional myosin, type 2 myosin which is responsible for muscle contraction, and there are unconventional myosin. There are 17 classes of unconventional myosins. There are type 1, type 3 to type 17. Type 18. So human possesses approximately 40 different types of myosins from 11 different classes. So each myosin performs specialized cellular function. So these image showing the microfilament myosin interaction. So these are the differences between the microtubules, microfilaments and intermediate filaments. These are their key features of distinguish these three types of cytoskeleton. This is the image showing the three types of cytoskeleton. So thank you my dear students for joining today's class. As we conclude today, remember that the cytoskeleton is the cell's dynamic framework combining structure with movement. Through the coordinated action of microtubule, microfilaments and intermediate filaments and motor protein, the cell maintains its shape, transports materials, divides accurately and performs specialized movements. Understanding this system is not just important for exam, it reveals how cells stay organized, responsive and alive. With this clear foundation, you are better prepared for undergraduate, postgraduate and NEET studies. So keep revising, stay curious and continue exploring the fascinating architecture of the cell. Thank you again.